This lecture is designated for 12th graders in AP Biology. This lesson will focus on the coronavirus, also known as SARS-CoV-2. The virus has had a major impact on these students in their lives, so it's important for them to understand the molecular biology of this virus and why it became such a society and scientific community issue. First, the students will be shown a video that shows the rate of infection between January 2020 and April of 2020. Following this video, they will receive this visual to explain the central dogma and how it is implicated in a virus. So first, the DNA is transcribed into RNA, and the RNA is then translated into a protein. And this visual is used to show them that the central dogma is very important in the spread of this virus. So after the first visual, the students will then be given this visual. So to explain, the spike gene in the SARS-CoV-2 genome encodes for the spike proteins. And these spike proteins are used to bind to a human receptor known as ACE2, which is how they infect the human cell. So with this S gene, it will use the central dogma, so going from DNA to RNA to translate it into a protein. And so when there is the S gene present, the spike proteins will be present. If for some reason this S protein is not available or it is taken out of the viral genome, no spike protein will be translated. Therefore, the virus will not be able to infect the human cell. Following the visualization of how the spike gene is translated into the spike proteins, the students will then be given this visualization of the SARS-CoV-2 virus attaching to a human cell, relying on the human nucleus to produce more of the viruses, and then how the virus will lyse from the cell to go spread into the rest of the body. These students will be given this visualization in order for them to understand that it is pertinent for the viral genome to contain the S genes so that the S proteins will be present in order for the virus to infect the cell. With this advancement in understanding the high affinity of the spike protein binding to the human ACE2 receptor, it has become extremely clear that it will be difficult to treat this virus. Following this lesson, students will be split into different groups. They will be given the hypothetical situation of working for the CDC. They will be tasked with figuring out how to stop the virus from spreading before it enters into the human body. Each group will be given scientific papers that explain CRISPR-Cas9, RNAi silencing, and mobile element insertions. Each group will formulate a plan in order to complete this task. The goal of this activity is for the students to identify the gene that is responsible for the spike protein formation and then figure out how to use the mechanism given to target this gene. After finalizing their plans to complete this task, they will present them to the class. Following this lesson and the activity, the students will be given a quiz. In order to track the students' understanding, the quiz will include questions about the central dogma, how the virus relies on host machinery to replicate and then infect, and how the spike gene is required in order to produce the spike protein. There will be questions such as, what is the process needed in order for DNA to become a protein? What would the SARS-CoV-2 virus look like if the S gene was not present? And what is one mechanism that could be used in order to prevent the S protein from forming? At the end of this lesson, students should be able to explain how the central dogma is implicated with viruses, how viruses rely on host machinery in order to replicate and spread, and they should be able to identify and explain the specific gene that is required for SARS-CoV-2 to infect human cells. This knowledge that they will be obtaining from this lesson will be extremely important for another pandemic or the routine spread of other viruses such as influenza.